It's okay. I'm on. I'm sorry, I didn't notice. You've already done it, and I'm recording now.
Okay, very good. Good. So at this point, then uh, we can begin. So, okay. So, okay, we're going to go from beginning. Okay. So, again, in my love of watching old movies and whatever, this I always consider to be the classic, apropos to what we're basically talking about today. In case people have not recognized it, the man on the couch is a guy by the name of Vincent Price, who was a star of many kinds of horror movies and whatever. And the movie in here is the movie called The Tingler. And the whole point of The Tingler was Vincent Price, as a mad scientist, wanted to basically see whether there were any kinds of changes that would happen under a state of extreme fear. So in this movie, what he basically does is he gets, of course, a woman who is mute, and he basically goes and he scares her, and he scares her to death. So now all of a sudden he then goes into the thing and starts to operate, and all of a sudden he exposes the spinal cord. But what you have here is not only a spinal cord, but a whole spinal column. And as we're going to learn today, we're going to be talking about spinal roots. So what you're looking at is a sort of centipede type of creature that is basically the mute woman's spinal cord that has gotten activated, and all of a sudden is starting to move all around the all around the room. And of course, the word "the tingler" comes from the tingling in your spine that you feel, let's say, under fear. And of course, being the mad scientist he is, of course, what part of the room does he go and run to? He runs to the part of the room that's furthest away from the door, falls over backwards onto the couch. The tingler jumps up on him and gets him. But in any way, I thought this is a very apropos kind of a picture where you're basically looking at things like spinal roots and whatever. So in a lot of what we're going to be basically covering today in a lot of ways is almost going back to that very first class where we were talking about phylogenetic development. And in phylogenetic development, you're looking at the development in the nervous system as one moves up the phylogenetic scale. And of course, at a very simple sort of situation, what you basically start off with is, as I pointed out, the earthworm, that is really a nervous system set with a whole series of ganglia that are interconnected with one another, in which one of the ganglia really does not have very much more power or whatever than another ganglion. But of course, a very important thing for the existence of the earthworm is for those ganglia to communicate with one another. And then to communicate, obviously, in the case of an earthworm, a whole bunch of somatosensory information as an extremely important part of what is being considered. So let's move on. So here what you're looking at is a spinal cord structure. So we're looking at, number one, the spinal cord and the spinal column. And what you're basically going to quickly learn in the next slide is that there are, quote, 31 segments to the spinal cord. The word segments is a misnomer. What it basically represents is the fact of the spinal cord, which is within the spinal column, and the spinal column is made up of a whole series of paired off vertebrae, of which there are 31 pair. And therefore, between each vertebrae is something called an intervertebral foramina, okay? And again, as we learned, a foramina is an opening, and in this opening, going either to the left or going off to the right, are going to be what we call dorsal and ventral roots, pairs of roots that are going out to the left and the right through all 31 intervertebral foramina. So one of the things that you can basically see here is that the spinal cord actually ends somewhere around L1, and that there's a very large space between the spinal cord proper and all the way down to the coccygeal segment. So if you remember in the last class, when we were studying the ventricular system, and we basically looked at a case like spinal meningitis, one of the ways that you would be able to look at spinal meningitis or assess it is to stick into a needle into this lower part of the subarachnoid space, which is around that spinal column, and well away from the actual spinal cord itself. So basically, when we're talking about these segments, we're talking about the most rostral segments, 8 cervical segments, C1 to C8, then 12 thoracic segments, T1 to T12, then 5 lumbar segments, L1 to L5, then 5 sacral segments, S1 to S5, and then a coccygeal segment. So that is just a rough approximation of the spinal cord. So we have the spinal column and the spinal cord with those segments, and the cord proper is extending from the foramen magnum all the way to the lower border of the L1 vertebra. So again, the segments are defined by the intravertebral foramina to the chorda equina, not the shape of the cord. The actual shape of the cord, and we're going to be seeing this, is having 2 enlargements, a cervical enlargement and a lumbar enlargement. So here we're looking again at the cord here, and in relatively quick things, we can basically see some of the very important types of functions that the different levels of the cord control. So you can basically see moving from C1, C2, C3 is very much controlling the upper neck, and then we come down to the diaphragm, and then we move out from C5 to C8, all the way out our shoulders, arms, out to the hands and fingers. Then we can see the upper thoracic cord, T2 to T7, is controlling many of our chest muscles, whereas T9 to T12 is controlling the abdominal muscles. So if we basically, again, think about the thoracic cord as a place where inputs and outputs are going from the spinal cord, we can pair that up with what? The 10th cranial nerve, the vagus nerve, which I basically pointed out was a mixed cranial nerve that has both sensory and motor components. And that the vagus, if you remember, had a supradiaphragmatic vagus, which basically controls respiration, and a subdiaphragmatic vagus, which again controls a lot of digestion. So what you can basically see is there's an interaction or dual innervation where the torso is getting innervated by the supradiaphragmatic and subdiaphragmatic vagus, as well as getting innervation from the corresponding thoracic cord. Then we see in L1 to L5, of course, is the leg muscles and the lumbar cord, and then the sacral is controlling the bowel and the bladder. So that's a very quick look at the spinal cord. So we now have 31 spinal nerves. So there's 4 nerves per segment. 2 are on the left, 2 are on the right, 2 are dorsal, posterior, 2 are anterior. Now, we can basically point out that the dorsal roots are sensory afferents, and we're going to speak about this a lot today. So the dorsal roots that are dorsal or posterior for all 31 spinal nerves are bringing in sensory afferents. And there are different kinds of sensory afferents, and we'll describe them. The ventral roots were traditionally thought to be purely motor efferents. But in actuality, there are actually a rather large amount of sensory afferents coming in from the ventral roots, as well as these motor efferents. So now you can ask a question, gee, why didn't they know that sooner? And the reason is that the ventral root motor efferents are heavily myelinated fibers. These are going to be the fibers that are going to be innervating the muscles and controlling things. And then what you basically then discovered was an enormous number of sensory afferents, which were unmyelinated or known as C-type afferents coming in. And the interesting thing there is that C-type afferents are very, very commonly associated with pain. So pain fibers will come in not only through the dorsal roots, but they'll also come in through the ventral roots and then go up into the dorsal horn. So each of these roots, 4 nerves per segment and everything, go through intravertebral foramina. So you have these 2 bony structures, a vertebrae and another vertebrae. And then what you have is cartilaginous tissue that separates those vertebrae. And then there's a series of blood vessels and, of course, the nerves that pass through the foramina. Now, the point of the matter is what is the nature of that cartilaginous tissue? It is disc-shaped. So what you basically hear with very often lots of things having to do with back pain is something caused by a slipped disc. So one thing can either basically the vertebrae can come out of alignment, or that the vertebrae, simply because of some injury or whatever, can wear down that cartilaginous tissue. And of course, then what basically happens, the vertebrae have a chance, especially with postural adjustments, to basically touch on one another and very often cause weird somatosensation, but very, very prominently pain. So the very nature of these spinal nerves, as they enter and exit the spinal column, become very, very important. Okay, all right. So now another concept here that is very important is that there is a dermatomal arrangement to the spinal cord and the spinal nerves. And this is going to become very important because we can go back to a finding or a theory all the way back in 1828, which was brought up by Muller, and Muller talked about the law of specific nerve energies. And he was quite prescient in making the argument that there were specific nerves for specific sensory events. So without knowing that there were things called rods and cones, knowing that there were hair cells, knowing that there were Pacinian corpuscles, Meissner's corpuscles, free nerve endings, knowing that there were taste buds and taste receptors, and then thousands and thousands of olfactory information that is done by specific types of nerves. He basically postulated this. Now, there's a second thing. The second thing is, if you basically remember in the first couple of lectures where I described about the precentral gyrus and the postcentral gyrus, that the precentral and postcentral gyri was somatotopically organized such that on the medial superior part of the head or the brain, you basically had the lower extremities, and as you moved out laterally and down the side, all of a sudden you moved up the body until you got to the head. So one of the things that this starts to suggest, and of course, neuroscience and psychology is filled with this, is a sort of vitalism. The whole argument that this is some sort of homunculus, and this homunculus basically is controlling our somatosensory experiences and motor experiences. The point in that basically is a classic top-down type of neuronal organization, that it is the cortex that's running everything, and everything else just flows from that. It's actually the opposite. It is a bottom-up type of situation, because what we're going to be learning today, as we look through the spinal cord and we look at the somatosensory types of inputs that are coming in, they're going to be coming in to specific parts of the spinal cord, such that, as you can see, C1 through C8 is around the head and the neck and the arms. Then, other than the upper thoracic controlling a specific part of the frontal distal arm, it basically is controlling the trunk. Then L1 to L5 is getting inputs and sending outputs to the pelvis, hips, and legs. And then sacral is controlling, is controlling the bladder, the anus, and the outside leg. And of course, the coccygeal is the tail vestigial, and about the only creature that I know that had a human-like creature, again in the movies, that had a great tail, was the infamous Barf in Spaceballs, played by John Candy, walking around with his tail knocking things all over the place. Okay, so there's a dermatomal arrangement, and you can see this dermatomal arrangement both on the posterior and anterior surfaces of the body. So now let's start to look at these segments. And we're going to be talking about the cervical segments, the thoracic segments, the lumbar segments, and the sacral segments. And the things we're going to be seeing with a cervical segment is that there is, first of all, an enormous size. It's very, very large. And that there's far more white matter than gray matter. So why is there such a large amount of white matter? So at this point, I'd be looking around the class. Everybody would be looking at each other or looking away and not answering. So I asked my rhetorical question, and now I'm going to give you my actual answer. And the reason for this is that the somatosensory paths that are coming up from the lower levels of the spinal cord on their way to the brain have to traverse the cervical segments. And conversely, all of the descending tracts coming out of the brain going down to the spinal cord and to lower parts of the spinal cord also have to traverse this cervical segment. And in general, the cervical segment has an oval shape, sort of like that. Now, the other thing that we're going to be basically looking at very much today is a system called the dorsal column system. And the dorsal column system is a somatosensory system which is transmitting information from the periphery through the spinal cord right up to within a few synapses to the postcentral gyrus. We're going to learn certain aspects about this system. That number one, that a vast majority of the fibers that are ascending never synapse in the spinal cord. They literally enter the spinal cord at the level that they're at, and then they shoot upwards. The first place where they synapse will be in the caudal medulla. So if you go back to that second lecture, when we were looking at the dorsal surface of the brain without the overlying cerebellum or cortex, what we saw in the caudal part, right around the obex where the fourth ventricle ended, we saw these four protuberances called the nucleus gracilis and the nucleus cuneatus. The fibers that innervate the nucleus gracilis and nucleus cuneatus are coming up from the spinal cord, and they are then called the fasciculus gracilis, which are two medial, and the fasciculus cuneatus, which are two lateral. So the point of the matter is, what the word fasciculus is describing is a large fiber tract. And the fasciculus gracilis, that what we're going to see here, is actually fibers coming in from the sacral, the lumbar, and the lower thoracic segments. The fasciculus cuneatus is going to be made up of fibers coming in from the upper thoracic and cervical segments. So if you remember, I said that information is bottom up. So what basically happens is these fibers will immediately come into the spinal cord and then ascend up that midline. So which are the most caudal fibers? The sacral fibers. Then what will happen as they go up, and we now go past the lumbar segment, the lumbar fibers come in and join them just laterally. Then the lower thoracic fibers come in and they join. Then the upper thoracic fibers come in and they join. And then finally the cervical fibers come in and they join. So this fasciculus gracilis, which is primarily made up of very medial sacral fibers, then lateral to that, lumbar fibers, then lateral to that, lower thoracic fibers, and the fasciculus cuneatus, which has upper thoracic and then cervical fibers. They all come up in this very somatotopically organized type of fashion. So, and we're just going to look at this and do this over and over again through the as we go through this lecture. So, if we look at these cervical segments, and here is a sort of a rendition of a C1, C4, and C8 segment, what do we start to first of all see? What we do see is that these cervical segments are larger than their counterparts. And then what they have is far more white matter than gray matter. And in all of the segments, what do we have? What we have is a dorsomedial septum. That is an opening. Think of it in very much the kind of way as an interhemispheric fissure, but it's called the dorsomedial septum. And you can see that in all of the sections that we have. And then you have an anterior medial septum right here. Okay. And then in the cervical level, but also in the upper thoracic level, what do we have in addition to a dorsomedial septum? We have two dorsolateral septi. So the dorsomedial septum and the dorsolateral septi will form in it the fasciculus gracilis. There is the fasciculus gracilis. Lateral to the dorsolateral septi is the fasciculus cuneatus. Note that they are what? White matter. And it's these, it is this major somatosensory pathway. Then what you can see right here is you can see the dorsal horn, and you can see the ventral horn. And we'll talk about that more, and they're quite large. And then right in the center here is the central canal. And remember that the central canal is the continuation of the ventricular system. Remember, the ventricular system, as it went through the fourth ventricle, had four possible ways to go. One was to continue down into the spinal cord through the central canal, or there were two lateral apertures called the foramina of Luschka, or one medial aperture called the foramen of Magendie. So there is just a general quick idea of what the cervical segments are. Now we go to the thoracic segments. And the thoracic segments, there's still more white matter than gray matter. And why is there? Again, it is those fibers that are either ascending up to the brain or descending from the brain to the lower parts of the spinal cord that have to traverse the thoracic segments to get there. So that's why there's that ratio. And then in all 12 segments of the thoracic cord, what we have is something called a lateral horn. So in the previous slide, I pointed out to you that we had a dorsal horn and a ventral horn, dorsal horn and ventral horn, dorsal horn and ventral horn. Now when we look at the thoracic segments, we see a dorsal horn, a ventral horn, and a lateral horn. A dorsal horn, a ventral horn, and a lateral horn. Now, why is that? It's because at the cervical, lumbar, and sacral levels, what we are getting is somatosensory inputs, okay? Whereas in the thoracic cord, in addition to getting somatosensory inputs, what we are also getting is viscerosensory afferents and visceromotor efferents. And what does viscero mean? That basically, when you think about, you're thinking about the type of innervation. A somatosensory afferent is coming from either skin or striated type of muscle, whereas viscerosensory afferents and visceromotor efferents are dealing with a lot of smooth muscle, which of course will be in the chest and will be in the digestive system. The other thing, so the lateral horn is always seen there, and then in T10 to T12, what basically starts is right down around here, and here, what you're basically going to have in the intermediate part of the spinal cord is something called the columns of Clark with an E. And it is here where spinocerebellar input comes in. So again, remember the hyphen. Spinal is information from the spinal cord going into the cerebellum. And these columns of Clark are going to be providing sacral, lumbar, and thoracic input into the cerebellum, in addition to doing whatever it's going to go in terms of spinal input into the brain for somatic information. Now, what we obviously with 12 thoracic segments, it's hard to differentiate, but we can differentiate the thoracic segments, whereas all of them have the lateral horn, but the rostral six, T1 to T6, still have a fasciculi gracilis and cuneatus, whereas T7 to T12 only has a fasciculus gracilis. So if we go back and look at this, here we saw in the cervical level, the dorsomedial septum, the dorsolateral septi. So we have a fasciculus gracilis and a fasciculus cuneatus. In the upper thoracic segment, admit her, there we go. In the upper thoracic segment, we have a dorsomedial septum and dorsolateral septi. So we still have a fasciculus gracilis and a fasciculus cuneatus. But in the lower thoracic segment, notice here we just have a dorsomedial septum, so hence only a fasciculus gracilis. And again, why is this? It's because as fibers are being added into this dorsal column system that comprises the fasciculus gracilis and fasciculus cuneatus, as we are caudal, it'll only be sacral fibers. Then as we move up to the lumbar level, it'll be sacral fibers medial, lumbar fibers lateral. Then we move through the lower thoracic, and we then have sacral medial, lumbar, then lower thoracic, upper thoracic. Now the upper thoracic is adding into the lateral fasciculus cuneatus, and then the cervical will add from there. So that is basically why we have these why this is happening. So here we go. Now we look at the lumbar and sacral segments, and now all of a sudden we flip in that gross way of looking at the spinal cord, in that there is far more gray matter than white matter. Why? Because a lot of the fibers that were entering in the spinal cord going to the brain have already gone through, other than the local lumbar and sacral, and the descending ones have sloughed off. So the lumbar has a circular shape, and it has very large anterior and dorsal and ventral horns, or anterior and posterior horns. L3 to L5 really control all of the large muscles in the lower extremities. The sacral is a small size and has the largest gray to white ratio. I keep asking why, so you can remind yourself already, because a lot of that afferent and efferent action between the spinal cord and the brain have already occurred. So there is there's a sort of a gross view of spinal segments, and you can actually, looking at a spinal segment, start to identify it next to each other in terms of these very, very gross differences. Okay, so now when we are going to be looking at spinal sections, we're going to be looking at a whole bunch of these things, and I'm just going to go through each one of these terms. So the first thing I want to talk about is the dorsal root ganglion, or DRG. And of course, on either side of the spinal cord, lateral but off of the spinal cord itself, is a dorsal root ganglion for each one of these nerves. And what is the dorsal root ganglion? Notice the word ganglion versus nerves or cells. Ganglion is usually attributable to the peripheral nervous system. So now let's think about a dorsal root ganglion. What it is a collection of cells that are coming on the right side of various types of inputs. And they're a collection of cells. Well, if it was just a bunch of cells and we were dealing with the classic learning of what a neuron is, you would say, all right, these cells are getting receptors on their cell body, and then they have dendrites and they get inputs there. Well, how the hell then do we get information from the tips of our fingers if our dorsal root ganglion is way in here? And the reason is because these DRG cells are highly specific. What they do is they have virtually no dendrites. And what they do is the cell sends out an axon, and the axon bifurcates. So it goes in one direction, going out and innervating a place out there on the periphery. It could be on the dermis, it could be in the epidermis, it could be on a muscle, it could be on a tendon, it could be on a number of things. And then there is the ending there is going to have a certain kind of property that we're going to talk about later that's going to take in some form of somatosensory, or in the case of the thoracic cord, viscerosensory input. And then that type of input will then shoot along the axon. It goes right past the bifurcation and goes into the second part of that axon that is now entering into the spinal cord. And we're going to have two kinds of places that those fibers can go. What they could do is those fibers could go enter into the spinal cord and then shoot up either the fasciculus gracilis or fasciculus cuneatus through the dorsal column system. So in that case, those particular cells are not going to synapse until they get to the medulla. The second group is the axons come in, And what they now do is innervate one of the 5 major areas of the spinal cord laminae 1 through 5, which I'm going to basically go through. So the dorsal horn itself has a gray matter cells, and of course it will then have axons that'll go elsewhere, and we'll talk about them. Then also in the dorsal horn, you also have the dorsal columns with the fasciculus gracilis medial, the fasciculus cuneatus lateral, and then what we have on the very extreme level of the dorsal horn, is a thing called Lissauer's tract, which is fibers. And what you can basically think of is an interaction between lamina 1 of the dorsal horn and Lissauer's tract. And we can now go back to our friend the earthworm. Remember, the earthworm had a pair of sets of ganglia, and these ganglia would communicate with one another to allow that earthworm to do whatever movement it went through and whatever. In a lot of ways, Lissauer's tract and lamina 1 of the spinal cord acts very much like an earthworm type of situation. It's spino-spino communication. So Lissauer's tract is communicating among different levels of the spinal cord, up a few segments and down a few segments, okay? And we'll talk more about that. Now, so we just covered the dorsal root ganglion. We sort of post-dorsomedial septum, dorsal lateral septum, the dorsal horn, the dorsal columns. Now we move into the center part of the spinal section, which is called the intermediate zone. And this has a lot of gray matter. So right down the center of that intermediate zone is going to be the central canal, which of course has CSF. And then there is going to be an area, gray matter, all cells, called the pericannular gray, which immediately surrounds the central canal. Now, what I want you to think of is go back in your mind, go rostral and go to the 4th ventricle. And we will have a periventricular gray in the medulla and the pons. When we go up into the cerebral aqueduct, we're going to have immediately surrounding the cerebral aqueduct in the midbrain, the periaqueductal gray. And then if we go rostral to that, we have the 3rd ventricle, which is vertically aligned, and we have periventricular gray, which are hypothalamic tissue. That periventricular gray hypothalamic tissue, periaqueductal gray, periventricular gray of the pons and medulla, and the pericannular gray around the spinal cord are among the most phylogenetically old cells, ancient cells in the human brain. And what we're going to get from this, I'm not going to go into it in detail now, but what we basically can begin to talk about is very basic homeostatic functions that these areas play roles in. So they're going to be playing roles in feeding. They're going to be playing roles in drinking. They're going to be playing roles in sex. They're going to be playing roles in aggression. And they're going to be playing roles in survival and stress. And we'll just see how all of that works with that phylogenetically old part of the brain, because those are the 5 functions that any animal organism needs to do in order to keep going. Then we are then going to look at lateral to the intermediate zone. We have what we call the lateral columns, which are all fiber tracts. So what we're going to have here is the lateral corticospinal. Notice that's a descending. Lateral spinothalamic, that's ascending. Spinocerebellar tracts, those are ascending. And they're all white matter. Then finally we get into the lower part of the spinal section, which is the ventral horn, which is gray matter. We're going to be looking at all of these things. I'm going to be identifying them. And the ventral columns, which are going to be the anterior spinothalamic and anterior corticospinal tracts. And then finally, just like we paid attention to the posterior median septum, we'll look at the anterior median septum. And the important thing in looking at things like the dorsal column system, looking at the lateral and anterior spinothalamic tracts, what we're going to see is this topographical organization. That is where the fibers are within this system will identify from what part of the body is this information coming from. So that is, again, a very important type of thing to understand. So here, just looking at this sort of schematic drawing, we can basically see this general organization. So we can see the dorsal root ganglion, and then the dorsal root ganglion is entering into the spinal cord, and it's going to do one of two things. It's going to enter medially and go right up the dorsal columns, or it's going to enter in more laterally and innervate the first 5 laminae of the dorsal horn. Then what we also see is the dorsal columns and the dorsomedial septum. We see the central canal. And then what we see is the intermediate zone of the spinal cord. And then we see the ventral horn, and then the ventral roots, which are out. But like I told you, there's a certain percentage of especially unmyelinated fibers that also enter this way and innervate the dorsal horn. So what you basically can see is, think of this as an analog clock, and think of the dorsomedial septum as 12 noon, and think about the anterior white septum as 6 o'clock, and then think of the center part of the clock being virtually all cells, whereas on the outside we have a series of fiber systems that are starting at 12 o'clock and going to 6, and then replicating themselves from 6 o'clock going back to 12. And we'll be identifying them at each of the different sections. So you can see the gray matter and the white matter. By the way, I'm not seeing my chat thing. I'm assuming, so let me just stop there for a second. Do people have any questions? Okay. So here is a picture of a dorsal root ganglion. And here are dorsal root fibers going into, either going medially into the cord or going laterally into the laminae and innervating them. And again, these dorsal root, these cells that are out in the dorsal root ganglion are made up of cells that bifurcate. One end going, the axons are going out to the periphery and doing the innervation. The other part is entering into the spinal cord. Okay. So now, what are these DRG inputs and how can we divide them? Well, we can divide them into 2 general categories. And when I say these two categories, it's far more of one than another. It's not like every A alpha fiber does this, but the vast majority do that, and the A deltas and the Cs do that. So A alpha and A beta fibers are fibers that are that can be physiologically identified by a large amount, heavily myelinization, and therefore they are extremely fast conducting. And these A alpha and A beta fibers enter medially directly into the dorsal columns through the dorsal horn. And what do they typically modulate? They modulate fine touch, fine pressure, fine temperature, and vibration. The A delta is lightly myelinated. The C fibers are unmyelinated. So therefore, the A delta fibers are slower conducting, and the C fibers are slowest conducting. They enter laterally to the dorsal horn, and what they do is they synapse in somewhere between lamina 1 and lamina 5 of the dorsal horn. In addition, C fibers can enter laterally through the ventral horn. The A delta fibers are usually involved in gross pressure, gross temperature, gross type of touch. And then the A deltas and Cs are also very much involved in pain. So first of all, here is an enigma. If we basically go and we try to say, well, if I'm going to go and I'm going to design a nervous system, wouldn't it be important to have my emergency switch in the case of imminent danger where the organism could die or the organism can get horribly injured? Wouldn't that system be the fastest? And the point, and that would have to do with the perception of pain. And the point of the matter is, it's actually the slowest. And what we're going to be noticing is in these pain pathways are the most diffuse. And we'll basically go and build and talk about why that happens and why that is organized that way. So now what I want to cover is the spinal cell groups. If you remember, I used the word spinal laminae. What is laminae? L-A-M-I-N-A-E. What it basically means is layers. And again, the point of the matter is, for certain parts of the spinal cord, layers is a very appropriate way. One layer, then another, then another, then another, superficial going in. But then around lamina 6 to 10, that layered aspect breaks down. But we'll explain. So the person who probably did the best initial description of this was the Norwegian neuroanatomist Rexed. And I want to basically tell you that when you look at this and you think about a human being, the human being's laminae are a bit more complex. There's a lot of A's and B's in here and things like that. Why? Because what Rexed used as his subjects in studying these laminae were the fetal kitten. So the most superficial and the most dorsal of the laminae is called the posterior-marginal zone of the dorsal horn. And what it does is border Lissauer's tract. So these are these are cells that are getting input from somatosensory input at whatever level of the cord you're at. And that input is exciting lamina 1 neurons, and then those neurons, when they fire, will send axons out into Lissauer's tract, and what there is spino-spino communication. And basically what is happening is, depending on what's going on here and the extent of the stimulation that you might be getting along a particular area, that many levels of Lissauer's tract would get activated and whatever. So Lissauer's tract is very much involved in spinal communication, and the cells of lamina 1, the posterior marginal zone, are involved in that as well. Then now let's look at laminae 2 through 5. Lamina 2 is called the substantia gelatinosa. It looks gelatinous, okay? Lamina 3 is called the proper sensory nuclei. Lamina 4 is called the proper sensory nuclei, otherwise known as the nucleus proprius. And then lamina 5 is known as the medial and lateral reticular nuclei. Now, what you should be doing in general organization is pairing lamina 2 and lamina 5, and then lamina 3 and lamina 4. Why? Because the inputs that come into lamina 2 and lamina 5 from these laterally projecting dorsal root ganglion axons coming in, the vast majority of these inputs are C-type fibers relative to A-delta fibers. And in lamina 2 and lamina 5, the type of somatosensory input that comes in there is called nociceptive input, N-O-C-I-C-E-P-T-I-V-E, basically talking about pain. Now, when we go and talk about pain in animals, and I always remember my first general lecture in the fall of 1979, I started talking about pain fibers coming in because that was the major research I was doing at the time. And a sort of very pompous professor in my department and a colleague named Jack Orbach, who basically always walked around going, well, I had the unique ability to have worked with Carl Lashley and D.O. Hebb, two of probably the preeminent physiological psychologists of the early 20th century. He goes, Richard, it's not pain, it's nociception, okay? And the reason why you make the differentiation is, according to a guy by the name of Ken Casey, who did this very well, is that pain basically is looked at in terms of what? 3 major dimensions: a sensory discriminative dimension, a motivational affective dimension, and a cognitive dimension. Whereas nociception is that form of pain that is primarily nociceptive, sensory discriminative. So it is a substantia gelatinosa and medial and lateral reticular nuclei are bringing in these nociceptor information. We're going to go through this in a bit more detail. What's going to be important about this is lamina 2 and lamina 5 will give off axons, and they will form axons that form the lateral spinothalamic tract. Lateral spino-thalamic tract. So that lateral spinothalamic tract is carrying, yes, general somatosensory information, but it is a primary way of carrying nociceptive or painful information. And it's because those C fibers that were entering into the dorsal horn were synapsing more in 2 and 5 than in 3 and 4. So what is synapsing in lamina 3 and 4? What is synapsing primarily in there are those A delta fibers, those lightly myelinated fibers. And those, the axons of those fibers will synapse in lamina 3 and 4, and their axons will go and form the anterior spino-thalamic tract. So the whereas the lateral spinothalamic tract had as its beginning lots of C fiber nociceptive input, the anterior spinothalamic tract getting its fibers from lamina 3 and 4 is getting primarily gross pressure, gross touch, gross whatever. Now, lamina 2, 3, 4, and 5, in addition to forming the lateral and anterior spinothalamic tracts, will also send fibers into the dorsal column. So about 40% of dorsal column fibers are going to will have synapsed in the spinal cord, and it's the second order neurons that enter in there. We differentiate between the medial fibers that immediately went up and didn't synapse as dorsal column fibers. What we call these other fibers is dorsal column postsynaptic fibers. So there's DC fibers and then DCPS fibers. Okay. So now, when we get to the bottom of lamina 5, we now get to the base of the dorsal horn. And the base of the dorsal horn is going to become very interesting for us, especially as we reach the thoracic cord, because within the base of the dorsal horn, we're going to find the nuclei of Clarke, C-L-A-R-K-E. And these nuclei will give rise to the posterior and the anterior spinocerebellar tracts. And we'll talk a lot more about them pretty soon. Then we have an area called the zona intermedia, and it's exactly that. It's a sort of nowhere land between the dorsal horn and the ventral horn. And the zona intermedia begins to become very important, especially in the thoracic cord, where what? We're basically talking about viscerosensory inputs and visceromotor outputs, controlling the trunk and the torso and all of the interior systems. Then lamina 8 is basically the base of the ventral horn on either side. Okay? So it's going to be involved in motor. Then lamina 9, think of lamina 8 as the Pacific Ocean. That's lamina 8. Lamina 9, within the so-called Pacific Ocean, we will have these islands. And instead of having islands, lamina 9 are the large alpha and gamma motor neurons that can be found interspersed in the ventral horn that are controlling all of the extensor and flexor major muscles. So now think about this. We have a cervical cord, we have a thoracic cord, we have a lumbar cord, and we have a sacral cord. Where will we find far more lamina 9 neurons? We will find far more lamina 9 neurons where? In the cervical cord and in the lumbar cord, because what are we controlling? We're controlling our arms, we're controlling our hands, we're controlling our fingers, we're controlling our legs, we're controlling our feet, we're controlling our toes. I guess the only group of people that I would say might have very large alpha and gamma motor neurons might be belly dancers in a thoracic cord. But in most cases, we have this large distribution of these motor efferents in those things having to do with our major appendages. Then, of course, lamina 10 is, as I pointed out, in the center of the spinal cord is the central canal, and we have the gray matter surrounding that called the pericannular gray. So those are Rex's laminae. And here is, here's a whole bunch of things where we can look at lamina 1 through 6 to 7, and what we can basically see is in lamina 2, what we can basically see is mixed nociceptive sensory and other sensory information. Lamina 3 and 4 is cutaneous mechanical A beta fibers. Then lamina 5 is getting A beta, A delta, and C fibers. And then lamina 6 is now getting inputs from muscles, joints, and whatever. And lamina 6, of course, is going to be playing a role with the nucleus of Clark. And of course, what the nucleus of Clark is going to be doing is sending in cerebellar input. So of course, these inputs from muscles and joints and whatever is sort of telling us about the positional sense of the organism, something that the cerebellum basically needs to operate. Then what you can see is lamina 8 and 9 having diffuse responses and multisynaptic inputs there. So here is looking at sort of these A beta and A delta fibers coming into lamina 1 and then coming into 3 and 5. And you can basically see 3 and 4 are getting what types of information? They are getting information that has to do with basically gross touch, gross pressure, whereas lamina 2 and 5 are getting A delta and C type of information, which has more to do with nociception. And the C fibers, of course, will enter both through the dorsal and the ventral roots. So now what I want to pay attention to is to basically go and look at some spinal projections. So right here we have the cervical spinal cord, or a rendition of it, and we see the dorsal horns, we see the dorsomedial septum, we see the dorsolateral septi. So what you have is the fasciculus gracilis and the fasciculus cuneatus. And again, on this side, as I try to show you, is that as the fibers way down in the caudal portion of the spinal cord, the information comes in and ascends medially, and then any additional information is put on lateral to that. So what you can basically see is for the fasciculus gracilis, you have sacral information, lumbar, and then the lower thoracic is on either side, and then the upper thoracic and the cervical are more lateral in the cuneate fasciculus. These things project directly into the lower medulla, right behind the obex, of the first synapse going into either the nucleus gracilis or nucleus cuneatus. So now, if you remember, I said one easy way to think about the spinal cord is to think of it as a clock using the dorsomedial septum as noon and the anterior white septum as 6 o'clock. And what we can do is we can see a whole bunch of fiber tracts. Now, they listed 15 fiber tracts here. I am not going to hold you responsible for all 15. What I'm going to do is we're going to pick out the usual suspects. So what you can see is immediately lateral to the dorsomedial septum and bordered by the dorsolateral septi is the fasciculus gracilis. Then lateral to that, you see the fasciculus cuneatus. Then we see the dorsal horn. And of course, out here on the posterior marginal zone, not labeled, is the zone of Lissauer. Now, I'm going to skip number 3. I'm going to go to number 4. Here is the tractus, the lateral corticospinal tract. It's here and it's here. It's basically sitting just lateral to the dorsal horn proper. But what is the lateral corticospinal tract? But the lateral corticospinal tract is the vast majority of corticospinal fibers that decussated across the pyramidal decussations on their way down to the spinal cord. So remember, information coming from the post-central gyrus and going down into the spinal cord go via the corticospinal tract, and at the caudal part of the medulla, 95% of those fibers decussate and they form the lateral corticospinal tract. 5% do not decussate, and we'll get to that in a minute. Now, where we saw the lateral corticospinal tract, now let's look at number 5. Number 5 is the lateral spinothalamic tract. So whereas the corticospinal tract is going what? Descending, the lateral corticospinal tract is ascending. Where are these fibers coming from? They are predominantly coming from cell bodies that are in lamina 2 and lamina 5 that have gotten gross touch and nociceptive information. Now, let's make sure that we know something here. Whereas the fasciculus gracilis and the fasciculus cuneatus are ipsilateral to where the fibers came in, the lateral spinothalamic tract, these fibers are contralateral to where they came in. What happened? Well, at a lower spinal level, the fibers coming in from the dorsal root ganglion that were A delta and C entered laterally and innervated lamina 2 and 5. Then the axons of the cells of lamina 2 and 5 form the lateral spinothalamic tract, and either at the level of the spinal cord where they were or a couple of levels up, these fibers will literally decussate, and they will decussate, and we're going to see this over and over again. They're going to decussate at an area just below the central canal. Can't get my point, there it is, called the anterior white commissure. So the anterior white commissure carries lateral spinothalamic fibers. Okay. And then they shoot out over here and ascend. So now what I want to pay attention to is not number 9. Where are you? number 15, which is the tractus spinothalamicus anterior, or anterior spinothalamic tract. Where did these fibers originate? These fibers originated in lamina 3 and 4, where they got a delta input coming in, and the axons of lamina 3 and 4 also decussate across the anterior white commissure to form the anterior spinothalamic tract. So the lateral spinothalamic tract, number 5, and the anterior spinothalamic tract, number 15, are fibers carrying information from the dorsal horn, and they are now contralateral to where they originated, whereas the fasciculus gracilis and fasciculus cuneatus had fibers come directly in, and they haven't decussated yet, okay? So now let's go and pay attention. So that those fibers, 1, 2, 4, 5, and 15, all are at all levels of the spinal cord, and I want you to know them. Then the last one that I want you to know in here is number 9. Number 9 is the tractus corticospinalis anterior, or anterior corticospinal tract. Look at where it is. It is basically sitting right on the ventral surface of the spinal cord. And if you continued all the way up and you went into the brain, what would you basically run into? You'd run into the pyramids, wouldn't you? And remember what I told you about the pyramids. 95% of the pyramidal fibers decussate and form the lateral corticospinal tract. 5% do not decussate, and they continue in their path. So it is the anterior corticospinal tract, number 9, has fibers that do not decussate, that are motor fibers of the corticospinal tract, and they will innervate all the different parts of the brain. So 1, 2, 4, 5, 15, and 9 can be found at all of the different levels of the spinal cord: cervical, thoracic, lumbar, and sacral. Now, what basically happened in the lower thoracic cord? What I told you is that the base of the dorsal horn, lamina 5, now at the lower thoracic cord, forms the nuclei of Clark. And the nuclei of Clark are getting information through the thoracic cord and into the lower cervical cord of input coming in from both visceral and somatic input coming into there. And what the axons of the columns of Clark form is 2 tracts. One is called the dorsal or posterior spinocerebellar tract, and you can see that here. And then the other is the anterior or ventral spinocerebellar tract. That's right here. And so what are these fiber tracts going? They're going from whatever level of the thoracic and lower cervical cord where you are, all the way up into the cerebellum. In fact, it's going to go. They're going to constitute, or one of them is going to constitute, a major part of the inferior cerebellar peduncle. We'll talk about that later. But why are there 2 of them? Well, the reason why there's two of them is that one of these fiber tracts, that is the posterior spinocerebellar tract. Number three is formed by an ipsilateral projections from the columns of Clark. It's formed by an ipsilateral connection from the columns of Clark. Whereas the anterior spinocerebellar tract is going to be formed from the contralateral columns of Clark, and it'll cross the anterior white commissure. Okay, so why would you have two fiber tracts coming out of the columns of Clark and going to the cerebellum? One of them which is ipsilateral and one that is contralateral. Well, your first statement might go, well, they're just innervating the two different sides of the cerebellum. It's an interesting thing. How does the posterior spinocerebellar tract go? It starts in the columns of Clark, shoots out lateral, shoots up, becomes the inferior cerebellar peduncle, and enters the cerebellum. What does the anterior spinocerebellar tract do? It starts in the columns of Clark, it crosses the anterior white commissure, it goes into the anterior spinocerebellar tract, so it is decussated once, goes up, goes past the medulla, goes past the pons, gets into the caudal midbrain, and now joins the superior cerebellar peduncle. And what does the superior cerebellar peduncle do? It decussates. So now we have a double decussation. So what do we end up with on the same side? And now it comes back down and goes back into the cerebellum. What does this let's do a thought experiment. Let us say there are two adjacent column of Clark nuclei that get identical input. One of them is going to form, the axons are going to go through the posterior spinocerebellar tract. So it's going to go out laterally, not decussate, go into the posterior spinocerebellar tract, shoot up through the inferior cerebellar peduncle, and get to a cerebellar target. Let's say we count that in terms of Mississippis. 1 Mississippi, 2 Mississippi, and just imagine it doing that. And it takes 8 Mississippis to get up there. Boom. Okay? Now, at exactly the same moment, the other cell with the identical input sends out fibers across the anterior white commissure. Then it goes into the anterior spinocerebellar tract. Now it goes up through the spinal cord, up through the medulla, up through the pons, into the caudal midbrain, then back across the back across the decussation, the superior cerebellar peduncle, and then comes back down into the cerebellum. And let's, in our imagination, imagine that it synapses on exactly the same neuron. But that took 15 Mississippis to do. What does this tell us about the cerebellum? And what does this type of input represent? Remember, it's identically the same input coming in from the spinal cord, but it literally does one at 8 Mississippis and another at 15 Mississippis. Well, very often, whenever you encounter a person who might have taken Psych 101 or something like that, and you basically do a Freudian word association task with the person, and you say cerebellum, and you basically wait for a response. And invariably, a very common response, if there's a one-word response to cerebellum, is coordination. Or someone might actually also say timing, because all of the cerebellar dysfunctions really show how we can break up timing and movement and whatever. So, what is time in terms of the brain? Well, time, we don't have a clock going on in there. Yes, we do have some cells like the suprachiasmatic nucleus that may play roles in circadian rhythms and everything. But this type of differential input can come in, and that posterior spinocerebellar tract could be signaling with a change in activity that starts the beginning of something, whereas the exact same stimulus that got to the columns of Clark and now did that more circuitous route across the anterior white commissure, up the spinal cord, up the medulla, up the pons, into the caudal midbrain, across the superior cerebellar peduncle, and back down, that might be the end point. And basically saying, here's the signal, here's the signal. So encapsulated within the beginning of this signal and this signal is some kind of neural equivalent to time. So again, we're looking at these projections, and we're going to be talking about these projections a lot as we move through the course. Okay, so now what we're going to do, and this is the first of a series of lectures in which we are going to do a sort of William Tecumseh Sherman march through the central nervous system. So we're going to start at a sacral level, and we're going to be marching through the spinal cord, going rostrally. And then in subsequent lectures, I'm going to be marching through the medulla, I'm going to be marching through the pons, and then I'm going to be marching through the midbrain until I get up to the rostral midbrain, where the brain is just too large, and that's where I start to split off into certain things. So we're literally going to be following what is in the brain as we move to what is in the central nervous system as we move from the sacral cord all the way up into the rostral midbrain. Let's take 2 minutes. I'm going to freshen up my coffee, and all of you can take a deep breath for a minute. And I'm going to put this up. Okay, I'm back again. And I imagine if we're recording, we should start the recording again. If not, that was fine. And so now what we're going to be doing is covering each of these sections. So the first section that I'm going to be dealing with, of course, is a sacral section. So here are some of the things that we're going to identify here. The dorsomedial septum, the fasciculus gracilis, the dorsal horn, the lateral corticospinal tract, the lateral spinothalamic tract, the ventral horn, then the anterior spinothalamic and anterior corticospinal tracts, and then the anterior median septum. Now, in these series of sections, as we move caudally to rostrally, what I'm going to be basically doing is varying the type of section you're going to be looking at. Because in all of these sections, there has been a stain to magnify a certain characteristic of the cells or fibers that are found in that stain. The most classic type of cell stains that you can basically look at is something called a Nissl stain, Nissl. And what a Nissl stain stains is ribosomes. And where are ribosomes within a nerve cell? Where they are, 95% of them are found in a cell body. Okay? So therefore, when you stain with Nissl, you're preeminently staining cells, and they typically come out a purplish color. So now, even though, so the cells will now stand out, and the gray matter obviously will stand out versus the white matter. Then a second type of stain I do is called is a fiber stain. And the particular stain that we're looking at is called the Weyl-Weigert stain, W-E-I-G-E-R-T. And what a Weyl-Weigert stain stains is the myelin. So there, and the way it stains it is it stains it black. So now, when you're looking at a Weyl-Weigert stain, the areas that are the darkest is paradoxically going to be the areas where there's the greatest amount of myelinated fibers, and then the so-called gray matter will look paler than that. Okay? Now, first thing you're looking at is you're going, oh my God, there is, look at all of the things I'm going to have to identify. That's the bad news. The good news is that as we move up the spinal cord, a lot of these structures, a lot of these fiber tracts, a lot of these cell bodies are going to be common throughout the entire spinal cord. And then there are going to be places where all of a sudden things start to change. And that's the most important thing when you're basically looking at these, all of these sections. Because what you want to always ask is, all right, here's section A. Now I'm going rostrally to section B. What has stayed the same? And what is now different? And it could be two different kinds of difference. One difference is all of a sudden something that was there before is no longer there, or now something new is there. A third possibility, especially when we look at the brain, is we're actually going to see particular structures, quote unquote, move. Okay? And what they're doing is the fibers are going to their destination. Okay? The other major thing, again, is the importance of hyphenation. Whenever you see a hyphenated term, 99% of the time, what comes before the hyphen is where it originated. What comes after the hyphen is where you end up. So with that, let's go to first our sort of gross drawing, and then we can then look at an actual spinal section. So here we're looking at a sacral diagram, sacral cord. And then what you're also going to be doing when we look at the actual section, you have to realize that this is a Weil-Weigert stain. So therefore it's staining fibers. So therefore the fibers are going to look darker than the surrounding cells, okay? And on each one of these sections, there'll be the description of the level of the spinal cord you're in, then what type of stain you're looking at. So again, what are we looking at here? What we're looking at is the dorsomedial septum, and then the fasciculus gracilis on either side. Then lateral to that is we see a small dorsal root ganglion here and here. And then we have the dorsal horn. And what you can see in the dorsal horn is we have a rather large substantia gelatinosa, and then we have the proper sensory nuclei, and then we have the central canal, the intermediate zone, the central horns, and then we have the anterior median septum. So what should we be able to see in projections? Number one, we see the fasciculus gracilis, and it's only the fasciculus gracilis. Why? Because we're so caudal in the cord, nothing else is added in. So it is just the sacral fibers. Then we have the zone of Lissauer, which is spino-spinal communication. And now, what is on the lateral surface of the cord in terms of fiber tract? Here we have the lateral corticospinal tract, here and here. But if you looked a couple of slides ago, you saw in that schematic that there should be a posterior and anterior spinocerebellar tracts. But they're not at the sacral level. They only start at the thoracic level. So the most lateral projection here is the lateral corticospinal tract on either side. Then what we have is the lateral spinothalamic tract, here and here. Notice that the lateral corticospinal tract is lateral to the dorsal horn, and the lateral spinothalamic tract is lateral to the ventral horn. Then anterior to the ventral horn is the anterior spinothalamic tract, here and here. And then medial to the ventral horn and lateral to the anterior median septum is the anterior corticospinal tract, here and here. We have one more tract to talk about, and that is...
Sorry, Professor.
Yes? Right here is the anterior white commissure, right here. And what is the anterior white commissure carrying? It is carrying the fibers. It is carrying the fibers from lamina 2 and 5 on this side, crossing, so they form the lateral spinothalamic tract, and fibers from lamina 3 and 4, so they form the anterior spinothalamic tract. Those are the two tracts that cross the anterior white commissure at the spinal cord. Now, so there is the diagram. Now let's look at the actual section. So remember, it's a Wheel-Weigert stain. So what is staining here is extreme myelin. So what do you see as darkest right here? What you can see here is the fasciculus gracilis, which is heavily stained here because those are heavily myelinated fibers entering from the dorsal root ganglion, directly going in here and shooting up through the dorsal columns. Then what we can actually see, again with a Wheel-Weigert stain, is we can see on the superficial levels of the dorsal horn, we can see the zone of Lissauer. And the zone of Lissauer is being innervated by lamina 1, the posterior marginal zone, and is providing spino-spino information. Then what we see right here is the lateral corticospinal tract. Notice how small the lateral corticospinal tract is at this point. Why? Because so many of the fibers that were in the corticospinal tract have already left and innervated the cervical cord, innervated the thoracic cord, innervated the lumbar cord. Then we see a very well-formed dorsal horn and ventral horn. Now, these look very, very large. It's not because the dorsal horn and ventral horn are very large at that point. It's that the surrounding white matter is very small, okay? So we're looking at a small section that is dominated by, like I said, way at the beginning, more gray matter than white matter. So now what you have is the dorsal horn and ventral horn, and now lateral, there's the lateral spinothalamic tract. Here is the anterior spinothalamic tract, and within here is the anterior corticospinal tract. And then, of course, right here is the anterior white commissure, which is containing fibers from the lateral spinothalamic tract decussating and the anterior spinothalamic tract decussating. Now, there's something very interesting. If you're looking here in this Wheel-Weigert stain, what you see is quite a bit of white matter, both in the dorsal horn and in the ventral horn. Now, some of that white matter you can actually see moving towards the dorsal columns. So if you remember, I said the dorsal columns is made up of those heavily myelinated fibers that enter medially via the dorsal root ganglion and shoot straight up. But there's also a subset, about 40% of the fibers, that come from lamina 2 through 5 and form what is called the dorsal column postsynaptic system. So that's that. But now what we see is a rather large number of fibers that seem to move from the dorsal horn into the ventral horn. Now I go and ask my rhetorical question. What are those fibers that are moving from the dorsal horn to the ventral horn? Well, those are fibers that are going to be involved in some type of sensory motor integration that can be modulated by but does not need the brain. And of course, the thing that we're talking about is probably going to the very basics of the beginning of neurophysiology in the early 20th century with the classic work of Sir Charles Sherrington, Sherrington. And what did Charles Sherrington study? He basically studied neurophysiological interactions and neurophysiological events, and basically described things like the action potential and the refractory periods and the beginnings of resting potentials. And the system that he used was the simplest of systems, because what he studied was fibers that were involved in something called the reflex. So what we know is that we have many things called spinal reflexes. And again, that's somewhat of an oxymoron, because a reflex is something that usually does not involve the brain, so it obviously involves the spinal cord. So what in essence is a reflex? A reflex is going to have an input or a series of inputs that is then going to synapse in somewhere in the dorsal horn, very often in lamina 3 or 4, and then axons of lamina 3 and 4, in addition to becoming part of the anterior spinal thalamic tract or becoming part of the dorsal column postsynaptic system, some of the fibers go right down into the ventral horn and synapse in lamina 8 and lamina 9 especially. So you're synapsing in the base of the ventral horn or on the alpha and gamma motor neurons, and then that output goes and then moves a particular set of muscles. So what I suggest you do is look up classic reflexes and see that, but you can actually see these interconnections between the dorsal horn and the ventral horn. And of course, you can then modulate those things. And you can modulate them in one of two ways, either by doing descending inhibition onto the ventral horn neurons, or intercepting the message at the pass and synapse and doing inhibition in dorsal horn cells. So let me just give you a little example of that. I just want you to do again a thought experiment. You get your PhD. You get you do your postdoc. Now all of a sudden you got a faculty position, and we're not in the age of COVID. We're in the thing where people sort of get together. So here, somewhere in the first month that you have you're in your new position, and all of a sudden you get this invitation. Now an email invitation, it's not mail, okay? And it basically tells you you're invited to the president of your college or university's house for a meeting with new faculty members. So of course you want to be impress people and whatever. Now, you're in that situation, but you're also the weekend before in a situation where you went camping and you're sitting out by a campfire. So in that first situation, what you're doing is it's early morning, and you have made some coffee on the campfire, and you pour the coffee and you pour it into a metal cup, and that metal cup is sitting perilously close to where the campfire is. So all of a sudden you touch the metal cup, you're picking it up, and all of a sudden, the hotness of the cup, ouch, you take the cup and you fling it away, you're blowing your fingers and you're cursing like a sailor, okay? All of the motivational, effective, and sensory aspects of pain are going on there, and you got rid of the goddamn cup without having to go up into the cortex saying, cup is hot, it's burning your hand, drop the cup. No, you just did a reflexive act, okay? Now move a few days later, and you're at the presidential reception, and now all of a sudden a person comes over and they're standing there and they pour coffee into a cup, but the cup was sitting very close to wherever the heat source was, and you pick up the cup. The cup is exactly the same temperature as it was before, but now in this case, instead of flinging the cup away, blowing on your hands and cursing, you basically take the cup and you put it down. Why? Well, first of all, the sensory discriminative aspects of the pain, the motivational effective aspects of the pain are there, but there is also something else there. That is, there is a descending pain inhibitory system that engages sometimes cortical structures and limbic structures, but goes into the periaqueductal gray, then goes down into the medulla, and then goes down into the spinal cord, where what does it do? It specifically inhibits lamina 2 and lamina 5 neurons. It basically is taking those nociceptive pieces of information that are coming in, and you're inhibiting them presynaptically, so that now you don't engage that reflex and you put it down. Yeah, you might engage your brain, but you didn't break the president of your university's Wedgwood china, okay? So I'm just trying to give you sort of real things, and one can think of the many times where one might do a reflexive act, and in most cases it's a good act. You're blocking injury, you're blocking possible death. Now, if we switch ourselves from faculty members going to campfires or presidential meetings, we can also basically think about a situation in which we are now something like a chicken. And a chicken basically might learn, oh my God, anytime I'm getting endangered, I run away. However, there are situations in which the chicken recognizes that there is an imminent danger, there's an overwhelming need to move or run away, but yet you stay still. And you do it at 1 day of age. It's called tonic immobility in a chick. And why? Because that type of inhibition of these reflexive actions are there to preserve that creature, because the predator that's there, and you're not going to go interview the chick and say, how did you know it was a predator? The creature knows. We can use instinct, we can use all of the neural equivalents of that, but it basically says, don't do this reflexive act now. And there's certain situations in which that happens. But we basically see at a very elemental level some of the most simple sensory motor integrations that we go and look at. Okay? So there is a sacral slide. So now we look at the lumbar section, and we're going to see, again, the dorsal medial septum. We're going to see a larger fasciculus gracilis. I hope by now you know that. It's because the fibers that were already there are ascending or adding. Then we have the dorsal horn. We have a larger lateral corticospinal tract Why? Because all of a sudden, there's that many more fibers that have to still go down to the sacral cord, but the lumbar cord is enormous in terms of motor efference. Then this is going to be an upper lumbar section because we will see a nuclei of Clark. So therefore, we're going to see the posterior ipsilateral and anterior contralateral spinocerebellar tracts. We're going to again see the anterior white commissure and talk about the implications of decussation, the lateral spinothalamic tract, the ventral horn, the anterior spinothalamic, and whatever. So now what we're looking at here is a wheel, again, a wheel Weigert stain. One of the things that you can basically see is that there's far more white matter, and that the gray matter is basically staying the same, but it is getting dwarfed by the white matter. So again, we're in a lumbar. So we have the dorsomedial septum, we have the fasciculus gracilis, and if we were to evaluate this, we would have sacral fibers ascending, and then lumbar fibers lateral to that. We have the dorsal horn with the zone of Lissauer. We now have the lateral corticospinal tract, and we have the lateral spinothalamic. But now, because we have the nuclei of Clark that give rise to an ipsilateral posterior spinocerebellar tract, or a contralateral across the anterior white commissure anterior spinocerebellar tract, the most lateral portions of these fibers is the posterior spinocerebellar and the anterior spinocerebellar. Then lateral to the ventral horn is the lateral spinothalamic tract. Then inferior to ventral to the ventral horn is the anterior spinothalamic tract. And then medial to the ventral horn is the anterior anterior corticospinal tract. Then above the anterior median septum is the anterior white commissure. And the anterior white commissure in this case now has three major fiber tracts in it. It has the lateral spinothalamic tract, it has the anterior spinothalamic tract, and it has the anterior spinocerebellar tract. Then within the gray matter, what we have is again the dorsal horn. Then we have the base of the dorsal horn and the emergence of the nuclei of Clark. And then we have a large ventral horn. And of course, in this ventral horn, we're going to have more lamina 9 neurons in the sea of lamina 8, simply because we're going to have many more alpha and gamma motor neurons. So now, when we look at the lumbar cord here, what we can basically see is, first of all, a much, much larger fasciculus gracilis, the dorsomedial septum, the anterior medial septum. Then what we have is the zone of Lissauer. And then what we have here is the dorsal horn. Then we have the intermediate horn. Now we can't see this. We'll be seeing this in a second. This is where the nuclei of Clark would be. Then we have a very elaborate ventral horn. And if you notice in the ventral horn, we have far, far more myelinated fibers, because why? What is the ventral horn of the lumbar cord controlling? It's controlling all of the major hip, pelvic, leg, and foot muscles. So, and then we have an elaborated anterior white commissure. So then with the fiber tracts, we have the fasciculus gracilis, the zone of Lissauer, the lateral corticospinal tract, and the lateral spinothalamic tracts, both of them respectively lateral to the dorsal horn and lateral to the ventral horn. But then lateral to that is the posterior spinocerebella and the anterior spinocerebella. So again, in the magic things of a picture says 1,000 words, you don't want to define, you want to see where the lateral corticospinal tract probably is, like right here. Come on, where are you? There we are. Here is the lateral corticospinal tract. But just think of how you have to define it. The lateral corticospinal tract is lateral to the dorsal horn and medial to the posterior spinocerebellar tract. Or the posterior spinocerebellar tract is lateral to the lateral corticospinal tract. It sounds like Peter Piper picked a peck of pickled peppers, blah, blah, blah. Okay? And the same thing with our friend, the lateral spinothalamic tract, which is lateral to the ventral horn, but medial to the anterior spinocerebellar tract. Then we have the anterior spinothalamic tract. Then we have the anterior corticospinal tract, and then the anterior white commissure. Obviously, in many of these sections, I'm skipping a whole bunch of other tracts that you can basically see in there, but I'm trying to give you the major things where you can see what's common and continues, what stops, what begins, and exactly where. So now let's look at a lumbar section, but instead of looking at the lumbar section with a real Weigert stain, I am now looking at a lumbar section with a Nissl stain. And what does a Nissl stain do? It stains the section wherever there are ribosomes. And since 95% of ribosomes reside within the cell body of the cell, it is a cell body stain. So now we're going to really be able to start to see some of the laminae a little bit easier. So let's do that. And here we have a lumbar stain. So first of all, looking at that lumbar stain, notice again the absolute size of the fasciculus gracilis versus when we looked at that at the sacral level. I'm going to have to stop a second because I have one of my idiotic cats trying to get in the house. Come on in. There you go. Go ahead. I guess this whole experience humanizes us. This is the little prince. This guy's name is Welly. Okay, now go off somewhere. Okay, now, okay, he's going to come back and probably stand on me. But in any case, there is a very large nucleus gracilis. And then what you can see is a very nice dorsal horn right here in the zone of Lissauer. Here's the dorsal horn, laminae 1 through 5. Now you can see lamina 6. Look at these cells of the nucleus of Clark. They're very, very well stained, and they will give rise to what? The posterior spinocerebellar tract ipsilaterally, or the anterior spinocerebellar tract across the anterior white commissure contralaterally. So that is the nuclei of Clark. Also, what I want you to notice in the zona intermedia, you have that. But notice here is the very intensely stained pericannular gray, okay? Again, this is going to be very important when we discuss whole aspects having to do with stress and the whole sympathomedullary system involved in stress, which very much engages at the spinal level the pericannular gray, and we'll describe that. Then what I want you to look at is the ventral horn. And again, remember how I sort of poetically described it, is lamina 8 is the sort of ventral horn itself, which you can make akin to the Pacific Ocean. And then lamina 9 are the individual alpha and gamma motor neurons and the clusters of them that you can see very predominantly within the ventral horn. So that is a Nissl stain. And of course, what you should always be doing is sort of marching along here and again, using that 12-to-6 argument of the posterior median septum, the fasciculus gracilis, then the zone of Lissauer, then the lateral corticospinal tract, the posterior spinocerebellar, the lateral spinothalamic tract, the anterior spinocerebellar, the anterior spinothalamic, the anterior corticospinal, and the anterior white commissure. So now we move to a lower thoracic segment. And remember, we had 12 thoracic segments, but the easiest way to divide them is to basically look at T7 to T12 versus T1 to T6. All 12 thoracic segments have this additional lateral horn, whereas the lower thoracic segment only has the dorsomedial septum and therefore only the fasciculus gracilis. The upper thoracic segment will have dorsolateral septi that divide the fasciculus gracilis from the fasciculus cuneatus. So we're going to be basically seeing a dorsomedial septum, the fasciculus gracilis, dorsal horn, lateral corticospinal tract, the intermediate and lateral horn for visceral inputs, then the anterior white commissure, the pericannular gray, and then the rest of these sections. And here we're looking at now again a Nissl diagram. And in the Nissl diagram, again, we're going to be predominantly looking at cell bodies. So here we are. Now, again, realize that the relative size of the dorsal and ventral horns have not changed. What has changed here is the enormous amount of white matter, which is on passage ascending and descending fiber tracts. So what we can see right in here is the dorsal root ganglion. And then what we can see is see the dorsal horn that's relatively thin here. And then what we can see is the nuclei of Clark here and here. And then what we see right here is a very exaggerated lateral horn on either side with series of cells. So notice what the nucleus of Clark is going to be doing. The nucleus of Clark is going to be playing the major role in terms of spinal input into the cerebellum, integrating that information. Whereas the heavy amount of cells found out here in, let me just get there. Where the the hell did my, oh, there it goes. Out here in the lateral horn is what we have are cells in the lateral horn that are involved in viscerosensory information, information coming in from smooth muscles, and smooth muscles as variegated as the intestines, where you're doing peristalsis, to any other kind of smooth muscle, like lining the lungs and things like that, for any kinds of breathing, to visceromotor muscles, which are basically going out and innervating those muscles. Now, in addition, what we have again is in the thoracic cord, we have a very intensely stained pericannular gray, because in this case, what you're basically now dealing with is a whole series of sympathomedullary types of stress responses. So what you're basically controlling with those pericannular gray neurons that will eventually go out to the sympathetic ganglia, that then will then modulate the adrenals. It's very intense in the thoracic cord, and this will then modulate increased changes, let's say with stress, with respiration, and of course, the very powerful increase in blood pressure and increase in heart rate that's being modulated right here in the thoracic cord. Then we see the nuclei of Clark, and then what we can also see right here is, although there's not as many, there's quite a number of laminin 9 neurons in the lower thoracic cord, which again is controlling things like the diaphragm, et cetera, in terms of function. And so now let's go to an upper thoracic section. In the upper thoracic section, now what we pay attention to on that dorsal surface is no longer do we only have a dorsomedial septum, but we have two dorsolateral septi. And what are those two dorsolateral septi separating? It is separating the fasciculus gracilis, which is medial, from the fasciculus cuneatus, which is lateral. The fasciculus gracilis is going to have sacral information, lumbar information, and lower thoracic information. The emerging fasciculus cuneatus is going to have upper thoracic information shooting up towards the medulla. Then we have the dorsal horn. We have the lateral corticospinal and lateral corticospinal tracts. We have the posterior and anterior spinocerebellar tracts. We still have a lateral horn with the visceral inputs, and then the rest of these things. So now we're going to look at a Wheal-Weigert stain, which again stains fibers. And now look at it in the upper thoracic cord. So we're going to see the dorsomedial septum. Then we have the fasciculus gracilis, which has sacral, lumbar, and lower thoracic input. Then we have the fasciculus cuneatus. The most lateral portion is going to have upper thoracic input. Then we have the dorsal horn. We have the base of the dorsal horn with the nucleus of Clark. We have the ventral horn, and we have the intermediate zone and the pericannular gray. Then we have the anterior white commissure. So here on the projection system, we see the fasciculus gracilis, the fasciculus cuneatus, the zone of Lissauer, the lateral corticospinal tract, the posterior spinocerebellar tracts, the lateral spinothalamic tract, the anterior spinocerebellar tract, the anterior spinothalamic, and then the anterior corticospinal, and then the anterior white commissure. So here in its actual section,
Oh no.
Damn, we lost one.
Oh, he's gone. Oh.
Where'd he go?
I thought that was just me.
Me
Okay.
too. Oh my God.
There's
Can we-
Okay. All right. Santa, come back.
Who's gonna teach the class now?
Wait, isn't John the host? He's the one who lets people in.
No, but he stepped out.
Oh,
Remember?
John's in charge now.
like I got wiped out a second. Okay, I'm back. Good, I'm back. Okay. Woo. Okay, he's back. I see. All right.
I'm
A
back.
little bit of a relief. Okay. So let me go back to this. We're almost finished here. We'll be doing a lot of reviewing.
We missed you.
Oh, okay. Thank you. Here we go. God knows what happened. But anyway, so here we're in this upper thoracic and the actual slide. So again, you can see the dorsomedial septum. Very lightly, you can see the dorsolateral septi that separates the fasciculus gracilis from the fasciculus cuneatus. And we just sort of move along there. Okay, let me go to the next slide here. Okay, so now we're going to look at a mid-cervical section. So in this one here, we basically are going to still see the dorsomedial and dorsolateral septi that differentiate the fasciculus gracilis from the fasciculus cuneatus, the dorsal horn. But now, not only are we going to see the lateral corticospinal tract, but we're going to actually start to see the pyramidal decussation. Then we're going to see the lateral spinothalamic tract, then the spinocerebellar tracts, the ventral horn, and the rest. So now let's look at this mid-cervical wheel diagram. So first of all, just look at that diagram and look at the relative size of the fasciculus gracilis to this emerging fasciculus cuneatus, simply because we are creatures that are bipedal, we're upright, and we do a hell of a lot more with our neck, our shoulders, our arms, our wrists, our hands, and our fingers than we basically do with our hips and legs. So in a lot of ways, the massive amount of contribution and relative size of the fasciculus cuneatus is because of this massive cervical input. And then you also, as we went rostrally, we have this massive amount of the lateral corticospinal tract, which is much larger because there's going to be a bunch of neurons that are a bunch of fibers that are going to be exiting and controlling all of this motor function. Plus, the point is, very often when we describe things and we say, right here in the caudal medulla, that's where the pyramidal decussation starts. It's not, it's like moving a goddamn battleship. It's not, it just doesn't all of a sudden happen in one thing. It takes an amount of time. So we literally get down into the middle of the cervical cord, where we're literally seeing still decussations of these fibers. And what do these fibers have to do? They have to move from the ventral portion of the medulla, cross the midline, and then get out to the lateral corticospinal tract. So what they literally even do is go across and cut through what we would consider to be dorsal horn. Yet we live and we do fine, and we can see the size of the lateral corticospinal tract. And then, of course, we have still now, one of the things, we're in the mid-cervical cord. What we no longer have in the base of the dorsal horn is our friend, the nucleus of Clark. The nucleus of Clark started about L1. It persisted all through the thoracic cord, but then mysteriously around C8, all of a sudden, there's no more nucleus of Clark. So that constitutes a mystery for you, which I'm not going to solve for you for a couple of lectures. That is, by knowing the nucleus of Clark and knowing the posterior and anterior spinocerebellar tracts, what we're able to describe is how the cerebellum gets input from the sacral, lumbar, and thoracic cord. What it doesn't tell you is how the cerebellum gets information from the cervical cord. It does not come through the nucleus of Clark, which is no longer there. So, but we still have on passage the posterior and anterior spinocerebellar tracts. And we have the lateral corticospinal and the lateral spinothalamic and the anterior spinothalamic and the anterior corticospinal and the anterior white commissure. So now let's look at this wheel diagram, which again, remember, is fibers. So you have met my cat, Welly. I just said, one thing I always did as a thought experiment is I would describe the various cats I've had over 40 years and tell people that this is a paw print of a cat standing on the slide as the picture was taken. That is not. What this is right here and a little bit here is the actual fibers that if we thought ourselves more rostrally and we had the pyramids running, and then the pyramids then had to decussate. And what did they do? They're on the ventral surface of the brain, aren't they? Where is the lateral corticospinal tract? It is lateral to the dorsal horn. So what happens is as these fibers decussate, they also move more superiorly into that final position. So you literally can see some almost interruption of the dorsal horn by the decussation of the pyramids. So here is the dorsomedial septum, the dorsolateral septi. So you can see a very nice fasciculus gracilis. Look at the size of the fasciculus cuneatus. It's absolutely huge for all the reasons I said. Then you see the dorsal horn. Again, it looks smaller than some other things. It's not. It's just that there's just so much more. And by the way, remember I talked about the word oval looking? Look at how the shape of this of the spinal cord is in general. It's a sort of large oval sitting right up here in your neck. Okay, and then we have the decussation of the corticospinal tract. We have the lateral corticospinal tract. We have the posterior spinocerebellar. Then lateral to the ventral horn, we have the lateral spinothalamic and the anterior spinocerebellar. Then we have the anterior spinothalamic, anterior corticospinal, anterior white commissure. So now we now go up to the upper cervical level, and we have all of the things I basically just said before, but that much more in spades. So here we're up around C1, C2, and here we're looking at, again, a wheel diagram. And what you can again see is the amount of fasciculus gracilis, which is much smaller and dwarfed by the fasciculus cuneatus, simply because there's so much more cervical input coming in. And then what we see is even a more elaborated decussation of the corticospinal tract, pouring into the very, very large lateral corticospinal tract here. Then we have the posterior and anterior spinocerebellar. Then we have the lateral spinothalamic, anterior spinothalamic, anterior corticospinal, and the anterior white commissure. So when we look at this, now it really looks like my cat with both of its paws stepped across this, and that's the literal decussation of the pyramids, because as we're doing this cut, we're cutting, usually you're cutting coronally, but these fibers are going out laterally, and that's why you basically see this. So again, we have the dorsomedial septum, the dorsolateral septi, the fasciculus gracilis, the huge fasciculus cuneatus, the dorsal horn, the decussation of the corticospinal tract, the lateral corticospinal tract, the posterior spinocerebellar, the anterior spinocerebellar, the anterior, the lateral spinothalamic, the anterior spinothalamic, the anterior corticospinal, and the anterior white commissure. So, let me get to a larger thing here, and chat 2. He's back. Okay, very good. So I'm going to go at this point here, and basically, I know I went through a lot here. Notice what this particular lecture basically does. It was an enormous amount of information at the beginning, and I'm hoping that a lot of what I was covering by repetition and repetition and repetition, one can basically see you know, how these things elaborate as we move through the spinal cord. But the one thing that is in common here is, other than wherever on the dermatome you may be, the sacral cord, the lumbar cord, the thoracic cord, and the cervical cord act very similarly in terms of the nature of the types of inputs. So one major thing is that if we think at the level of the dorsal root ganglion, and we have particular cell types that are coming in, we notice very, very well is that the these very heavily myelinated sensory fibers, somatosensory fibers that are coming in, preferentially go and they move directly into the dorsal column, okay? And they go directly to the medulla. So one favorite thing that I always thought of is in the early 1990s, a sort of a basketball player came along who was utterly larger than life. And today he is omnipresent when that guy who was selling pizza, I forget, John, whatever the hell his name was, made a horribly insensitive racial slur. He got his butt pushed out of his own company. And who did they bring in but Shaquille O'Neal? Shaquille O'Neal sells you pizza. Shaquille O'Neal sells you insurance. Shaquille O'Neal sells you pain medications. And Shaquille O'Neal, for about 15, 20 years, was a very, very larger-than-life basketball player. So here's this guy, 7'3". So I just ask you to do again a thought experiment in which Shaquille O'Neal is lying on a Florida beach or a California beach, wherever you want to put him, and he's lying there, and somebody comes along and tickles him on his big toe, okay? Just a feather brush tickle on the big toe. So that sensory thing comes into his big toe, runs up Shaquille O'Neal's very long legs, runs into the spinal cord at about L3, and because it's tickling, it is A alpha types of fibers, heavily myelinated fibers. So now that still, remember, the cell is in L3 of the dorsal root ganglion. One part of the birth bifurcation is going all the way down to Shaquille O'Neal's big toe. The input comes in, passes the dorsal root ganglion, enters the spinal cord medially, and now starts to run up the rather substantial torso of Shaquille O'Neal all the way up through his neck, through the fasciculus gracilis, because it's of lumbar origin, and goes up and finally synapses on Shaquille O'Neal's caudal medulla in the nucleus gracilis. That is the dorsal column. That's the first synapse. But what have we learned, or what have you historically learned, is that information about that is lateralized. So what I didn't tell you is that I tickled Shaquille O'Neal's left big toe. So it's obviously staying on the left side until we get up to the nucleus gracilis and the caudal medulla on the left side. So now the second order neurons, the axons of the nucleus gracilis, is now going to carry that tickling information. What we're going to learn is that right at that point, those axons are going to go around the central canal, and that is where they're going to decussate to the right side. And then they're going to form a fiber tract. You don't have to write this down, I'm just describing it, called the medial lemniscus. And the lemniscal system, a lot of this stuff is borrowed from what? Classic psychophysics. If you went back into sensation and perception course, you probably have heard about a lemniscal system versus a protopathic system. A lemniscal system is a system really designed for fine-tuned information, where a protopathic information is much grosser. But this medial lemniscus goes up, and it goes to a thalamic nucleus called the VPL. And VPL stands for the location in the thalamus: ventral, posterior, lateral nucleus of the thalamus. And again, there is a somatotopic organization of the nucleus gracilis fibers and the nucleus cuneatus fibers that go into the VPL. And then that is synapse number 2. Then the information from the VPL will now go through the axons of the VPL, will now go through the posterior limb of the internal capsule and go right up to the postcentral gyrus and topographically innervate So if you remember where we talked about that homunculus, and we had what? On the superior surface of the postcentral gyrus, and then the lateral surface of the postcentral gyrus, we had an upside down person. We have Shaquille, we would be stimulating the superior and medial surface of the postcentral gyrus. On what side? Since we stimulated Shaquille O'Neal's left toe, it's going to appear on the right side. Now, if you looked at it from a cortex point of view, you would basically say, oh boy, isn't that cortex brilliant for figuring the thing out? No, it's a bottom-up system. It's the way the system has been organized and topographically laid out that basically informs us exactly where that feather had tickled Shaquille O'Neal. So this becomes a very, very, very powerful kind of system. And of course, what we're now going to go and do in the next lecture is start to go through each one of those projection systems. So the important thing for you to do from this lecture is, of course, I gave you the sort of background about spinal cord organization, but when you go through each of those slides, keep asking, what is there that has been there before? And then what is there that is now new? And then what is there that all of a sudden comes there and then disappears? So all the way through, we saw a dorsomedial septum. All the way through, we saw an anterior medial septum. Then we saw an emergence of a fasciculus cuneatus. Why? Then we saw an emergence of a dorsolateral septum. Why? Then we saw a nucleus of Clark. Then all of a sudden we didn't see it. And when we saw the nucleus of Clark, we saw the posterior and anterior spinocerebellar tracts. Then we see, as we move rostrally to caudally, we see the enormously large fiber tract start to get smaller and smaller. Why? Et cetera, et cetera. And this starts to give you a really good grounding. And then when you start to look and all of a sudden you see a hole somewhere sitting in a spinal cord section, you might then start to ask, gee, what was wrong with that person? Or you might see something wrong with a person and then ask, where would the hole be, or where would be the dysfunction? So here we are at 11:57. Does anybody have any questions, or does anyone basically want to feel my brain is full for another 3 hours and I want to take a break? So hopefully John Lyle and Allison Ferdinand has done a bang-up job of videotaping these things. I personally, I think there was the guy, Mr. T, I Pity the Fool, where he basically goes in the Rocky series. I don't know how you can watch me again. You must want to shoot yourself, okay? Or maybe when you're here the first time and you get it taped, when you get it taped, maybe you watch it later or something like that. But again, really go over this material, go over it yourself, hopefully go over it among people. And then at some point in the next couple of weeks, I'm going to have some asynchronous review sessions in which people can ask questions and whatever. So with that, I'm going to say bye and have a good week. And again, I'm going to do what I do now. Rather than going back to that recurring section and whatever, I'm going to probably the evening of Wednesday of next week, I will send out a new invitation and people can go there. Okay? So have a good week, everyone.
Yeah,
Bye-bye.
Professor, bye.
Professor,
Thank you, Professor.
Thank you.
I had a last minute question.
Oh, somebody had a last minute question.
I just thought of this. So when we say things like, it's probably a very basic question, but when we talk about the anterior and posterior, well, anterior corticospinal and spinal thalamic tract, could we just like say that interchangeably with ventral tracts?
Well, we're going to go through these things in a lot more detail in the next things and talk a lot and unite these things with function. So you're going to see. But look, when I say lateral spinothalamic tract and anterior spinothalamic tract, I'm clearly differentiating between the two, both as to where they are, but very much more importantly, what they do. So just like when I say, if I point in a thing and I point at our friend, the inferior frontal gyrus pars orbitalis, when you answer me, you're going to say inferior frontal gyrus pars orbitalis. You're not going to just say inferior frontal gyrus. That's not enough. And just saying pars orbitalis with nothing else is insufficient. You put the whole thing together. I'm sorry, sometimes the language is very, very long, but what we're really trying to do here is doing a visual auditory integration of all of this stuff. Does that answer your question?
Okay, I think so.
Okay, good. All right then. See ya.
Bye.
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 Bye.